Thank you very much. I'm so honored to be speaking in front of you, and so thank you all very much for coming and staying for my talk. I want to thank the JapanPrize Foundation for giving me this opportunity to talk about our work on cGAS, which is recognized by the JapanPrize. And I also want to thank Professor Okura for giving an excellent introduction to innate immunity, and especially the toll-like receptor pathway that detects pathogens outside our cells. And the pathway that I want to talk about is this cGAS STING pathway that detects pathogens in the interior of our cells. So this slide introduces three sort of major players in my talk. This is the DNA sensing enzyme cGAS, which is activated by binding to double-stranded DNA. And the activated enzyme converts GTP and ATP into this cyclic dinucleotide, cyclic GMP-AMP, we call it cGAM for short. This small molecule then functions as a second messenger that binds and activates this ER membrane protein called STING, which then activate a signaling cascade leading to an immune response. So basically, that's all you need to know about my talk. Okay, so this is the outline of my talk. First, I will briefly talk about how we discover cGAS and cGAMP using old-fashioned biochemistry, which I think is still very powerful. Then I will talk about our efforts in developing cGAS inhibitors to treat autoimmune and inflammatory disease. So why are we interested in recognition of foreign DNA? And that is because the recognition of this foreign DNA is one of the most fundamental mechanisms of host defense. Even bacteria can do that. And the classical examples are restriction enzymes that digest bacteriophage DNA or plasmid DNA. And the more recent example is the CRISPR system, which is the bacterial adaptive immune system that cuts viral DNA. In humans, DNA was known to stimulate immune responses for more than 100 years. When Nadia Mechnikov received his Nobel Prize in 1908 for the discovery of phagocytosis, he noted in his Nobel lecture that there are surgeons in France and Germany who introduce into the abdominal cavity either warm blood serum or nucleic acid with the object of bringing to the scene a protective army of phagocytes to ward the microbes off. So DNA was known to stimulate immune responses 40 years before it was known to be the genetic material. But we didn't quite understand how DNA stimulates immune responses until quite recently. So why do we want to have an immune system that detects DNA? First of all, this will be quite versatile, with the exception of RNA viruses, which are recognized by the RIG-I MAPS pathway that Professor Okura just mentioned. All microorganisms, including DNA viruses, retroviruses, fungi, and parasites, they all have DNA or require DNA in their life cycle. This then provides an almost universal mechanism to detect microbial infections. And secondly, the recognition of this microbial DNA or RNA provides a mechanism to detect those microorganisms that live and replicate inside our cells. We all know that toll-like receptors are very important in innate immunity, but toll-like receptors are membrane proteins with a ligand binding domain facing outside the cells. And as you just heard from Professor Okura, some toll-like receptors are localized on the endosomal membrane with a ligand binding domain facing the lumen of the endosomes, which is topologically equivalent to outside the cells. So toll-like receptors, by and large, detect pathogens outside of cells, and they are blind to those bugs that have successfully invaded our cells and replicated inside our cells. So we need another mechanism to detect these bugs. And the detection of this microbial DNA and RNA then provides a powerful mechanisms. When these bugs multiply the DNA and RNA, it's time to call in the immune system. But this detection mechanism has its liability because our cells have lots of DNA. And one solution to this problem is to keep our DNA out of cytoplasm. We keep our DNA in the nucleus and in the mitochondria. And inside the nucleus, we wrap our DNA around histones to form chromatins, which prevent immune activation. But this system is not perfect, and when the system fails, it can lead to a variety of autoimmune and inflammatory diseases, as well as cancer. Before I talk about this DNA sensing, I want to briefly mention the RNA sensing pathway, which my lab also worked on for many years before we got into the DNA sensing pathway. So when an RNA virus, such as influenza virus or Ebola virus or SARS-CoV-2 infect our cells, the viral RNA is detected by RIG-I or MDA5, which was discovered by Professor Takesha Fujita in Kyoto University. And RIG-I or MDA5 then activates this mitochondrial membrane protein that we call MAPS. We call this protein MAPS. It stands for mitochondrial antiviral signaling, and this protein was also independently discovered by 3 other groups, Professor Okura's group, who called this IPS1, Hongbin Su's group in China, who called this VISA, and York Sharp's group in Switzerland, who called this Cardiff. And we call this protein MAPS not only because it's on the mitochondrial surface, but also because we are in Dallas. And so in Dallas, we have a famous basketball team called Dallas Mavericks. And at the time when we discovered MAPS, Dallas Mavericks was playing against Miami Heat in the NBA final. So that's where we got the inspiration. So MAPS sits on the mitochondrial outer membrane, and then it activates these kinases in the cytosol, including IKK and TBK1. And IKK activates NF-κB, TBK1 activates IL-3. These transcription factors then move into the nucleus where they function together to induce type 1 interferons and other cytokines. And so that's how we fight RNA virus infection. And as I mentioned earlier, DNA can also strongly stimulate immune responses. And Ken Ishii in Professor Okura's lab and also Dan Stassen in Ruslan Mezhetov's lab show that the DNA in the cytosol can strongly induce interferon in a manner that is independent of toll-like receptors. So then the question is, what's the sensor that would detect this cytosolic DNA? And there has been a mystery for a long time. Downstream of this DNA sensor is this ER membrane protein called STING, which was discovered by Graham Barber in Miami and Hongbin Su in China. And STING can also activate IKK and TBK1 to induce interferons. So now the key question is, what is this DNA sensor? And several years ago, there was this intense race to try to identify the DNA sensor. And we joined this race by taking a different approach, and that is a biochemical approach. So in this type of biochemical approach, the first important step is to set up a cell-free system that recapitulates what's happening inside cells. So this is how we set up the assay. We took L929 cells, which have a functional STING pathway. We use shRNA to knock down STING, so we got rid of STING, hoping that we can trap a factor upstream of STING that is activated by DNA, and that potentially could include an activated DNA sensor. We then broke the cells open to prepare cell lysate, hoping that the cell lysate would now include an activated DNA sensor or an activator. We then deliver the cell lysate into another cell line, in this case THP1 cells, which also have a functional STING pathway. And we use a bacterial toxin called perfengrolysin O or PFO to poke holes on the plasma membrane such that we can deliver these factors into the cells. And STING is an ER membrane protein, so STING cannot get out of the cells. And when STING is activated, it will trigger a signaling cascade, leading to the phosphorylation and dimerization of IR3. And the dimerization of IR3 is the hallmark of its activation, and we can run a native gel to detect this IR3 dimer. So this assay sounds simple, but it took 2 years to get it to work. It was worked out by a talented postdoc in the lab, Josh Sun. So here, if we use DNA transfected extracts from DNA transfected cells, deliver the extracts into PFO permearized THP1 cells, now we can see this IR3 dimer. And these are just negative controls. So now we have the assay. And the plan was to start to fractionate these extracts to identify the activator. But before we did the fractionation, we asked a simple question. Is this activator a protein or not? So we took these extracts, heated up at 95 degrees to denature most of the proteins. We spun down the denatured proteins and took the supernatant and delivered into THP1 cells to see if it still has activity. And to our surprise, it still has activity. So this activity is resistant to heat treatment, resistant to proteinase K, resistant to nucleases, and is a small molecule. So these and other data led us to propose that perhaps the DNA sensor is an enzyme, and this enzyme can produce a small molecule that functions as a second messenger that bind and activate STING, that STING then activate IR3 to induce interferon. And so this model, which sounds provocative, turned out to be correct. And just to make a long story short, we identified this small molecule, which turns out to be a cyclic dinucleotide, cyclic GMP AMP. So this is GMP, this is AMP, and it has two phosphodiester bonds. One is 2' 5' link, the other one is 3' 5' link. So we call this 2' 3' cGAMP. And theoretically, cGAMP can have 4 isoforms. So this is 2' 3' cGAMP, and this one is 2' 2' cGAMP. This is 3' 2' cGAMP, 3' 3' cGAMPs. In collaboration with Heping's and Professor Chen's lab, we make all 4 cGAMP isomers and measure its binding affinity to STING. And it turns out that 2' 3' cGAMP, which is the natural product that we make in our cells, has the highest affinity. The KD is about 4 nanomolar. And if we deliver this 2' 3' cGAMP into cells, it can induce interferon beta with an EC50 of about 20 nanomolar. So it's very potent. So cGAMP is indeed a second messenger that binds to STING with high affinity and strongly induces the production of type I interferon. And now the question is, what is this enzyme? And this enzyme turns out to be very difficult to purify. We went to more than 2,000 dishes of cells, and we took different purification strategies, but we couldn't purify it to homogeneity. we could only achieve about 15,000-fold purification. And this is because this enzyme is really like low abundant. We estimated that we had to purify it to about 1 million-fold to achieve homogeneity. And so if we took our most purified fraction and run that on a gel and do a silver staining, it basically is a smear. And if we took this fraction, put it on another column, which is a disappear on the column, and we have to start all over again. So we're kind of stuck. Then we came up with a strategy. We reason if we have already purified this activity by 15,000-fold, and we have 1 million-fold to go, then in our most purified fraction, 1%, one out of 100 proteins, will be the protein that we're looking for. We just didn't know which one. However, if we can use mass spectrometry to identify all the proteins in different fractions, and if we can quantify the abundance of each protein, we reason the abundance of the protein of our interest must co-purify with activity. So my student, Jiaxi Wu, learned how to do mass spec and became very good at that. So he then identified 3 proteins whose abundance co-purified with activity. And one of these proteins was an unknown protein at the time that we named cGAS, or cyclic GMP-AMP synthase. And it has a nucleotide transferase domain which is homologous to another enzyme, OAS1, and the catalytic residues are highly conserved. So basically, this is how we discover cGAS and cGAM. We published these 2 papers back-to-back in Science in 2012. So one of these papers described the discovery of cGAS, the other one described the discovery of cGAMP. In fact, I submitted the cGAS paper from a hotel room in Tokyo when I came here for a conference, and it was accepted a month later. So Tokyo has been very nice to me. And these are the papers that basically brought me back to Tokyo. Now I'm standing in this podium. Okay, so fast forward to today, we have a much better understanding of the cGAS sting pathway. And so here I would use a few cartoons to illustrate this pathway. So here is a cell that is infected by a virus. So in this case, a DNA virus. And so the virus delivers DNA into the cells, and if some of this DNA end up in the cytoplasm, it's a danger signal that is recognized by cGAS. cGAS binds to the DNA and becomes activated. So this is the crystal structure of cGAS. And this is double-stranded DNA. So DNA binds to the cGAS, and it pushes this pink activation loop, and it induces a conformational change in the active site of the enzyme. And this DNA binding also induces a dimerization of the enzyme, which is very important for its activation. So this enzyme is now activated, and it converts GTP and ATP into this cyclic dinucleotide cGAM molecule that strongly stimulates immune response. Now, one molecule of the enzyme makes multiple molecules of this second messenger. So now the signal is amplified. And each of these second messenger will bind to a sting dimer on the ER membrane, and that changes the conformation of sting such that sting is now activated. Sting then activates the transcription factors NF-κB and IL-3. These transcription factors move into the nucleus to turn on the expression of hundreds of genes. They are important in immune and inflammatory response. Now, the cell biology of this cGAS sting pathway is also very interesting. So here is a movie recently taken by my postdoc, Ethan Parrots. But before I show you the movie, I want to orient you as to what to watch for. So when cGAS encounters DNAs, it undergoes this so-called liquid-liquid phase separation that this sort of formation of these liquid droplets in which cGAS is activated. Then cGAS then produces this small molecule, second messenger cGAM. cGAM then binds to sting on the ER membrane, and once sting binds to cGAM, then it traffics from the ER to the Golgi, where it activates IKK and TBK1 to induce NF-κB and IR3. So basically, in this movie, you will see that cGAS will form a puncta structure, basically liquid droplets, after it encounters DNA, and this is followed by the formation of the sting puncta structure that leads to activation of the downstream kinase. So Ethan took a human microglia cell line called SMC3. He labeled the cGAS protein with red fluorescent tag, sting protein with a cyan fluorescent tag, and then he just delivered DNA into the cells. And so the red signal is cGAS, and when it encounters DNA, it forms this puncta structure followed by the formation of this sting puncta structure. So this cGAS phase separation followed by the trafficking of sting from ER to Golgi, where it activates the downstream kinase, and sting is then degraded in the lysosome. And this is how you shut down the pathway to prevent overactivation of the pathway. Okay. And so this is another view of the cGAS sting pathway, and I will not get into the details of this pathway except to make an important point here, and that is cGAS is activated by any double-stranded DNA independent of the DNA sequence. And the crystal structure of the cGAS DNA complex shows that cGAS binds to the sugar phosphate backbone of the DNA. It does not have direct contact with the base pair. And that explains why it's DNA sequence independent. And this sounds boring, but it's actually very important because this property allows this single sensor protein to detect a large variety of pathogens as long as these pathogens contain DNA. And work from our lab and other labs have shown that cGAS is indeed a sensor for DNA viruses such as herpes virus, retroviruses such as HIV, bacteria such as MTB, parasites such as malaria. And another source of DNA is actually our own DNA. Because the DNA sequence independent, our own DNA can also activate cGAS. And as I mentioned earlier, our own DNA is usually kept in the nucleus in the mitochondria, but under some pathological conditions, our own DNA can get into the cytosol to activate cGAS, and in this case, it can cause autoimmune diseases. And one example of the autoimmune disease is called Akati-Coutier syndrome, or AGS. So these are the children with the disease. It's a very painful disease. So when these children are born, they produce a lot of type I interferons that damage their organs, including the brain, as shown here. And these are monogenic diseases, meaning that the disease is caused by mutations of a single gene. And now there are about 10 genes whose mutations are known to cause AGS. Most of these were identified by Yannick Crow in UK. And most of these mutations lead to the activation of the cGAS sting pathway. For example, some of these children have this disease because they have mutations in a gene called TRAX1, which encodes a nucleus that normally digests DNA in the cytoplasm. And the loss-function mutations of TRAX1 cripples the activity of this enzyme, such that cytoplasmic DNA accumulates, and that leads to a strong production of type I interferons that damage their organs. And work from our lab and other labs has shown that TRAX1 causes this disease most likely by activation of overactivation of cGAS. And this is shown very clearly in TRAX1 deficient mice. So these TRAX1 deficient mice die quickly after birth, so within weeks or months after birth, because they have very severe inflammation in many different organs. We asked, if we remove cGAS from TRAX1 deficient mice, can we rescue the disease? So my former student, Da-Xin Gao, generated TREX1 cGAS double knockout mice. Now 100% of the mice survived, and they didn't have any symptoms of disease. And interestingly, even if we just remove one allele of cGAS, so we reduce this protein by 50%, we can largely rescue the survival. And we think that this haploinsufficiency of cGAS in causing disease is due to this liquid-liquid phase separation that I just mentioned. So my former student, Mingjian Du, found that cGAS contains multiple DNA binding domains, including a disorder domain at the N-terminus. So cGAS can bind to DNA through this multivariant interaction. And the multivariant interaction between cGAS and DNA drives this phase separation that leads to the formation of liquid droplets in which cGAS is activated. And this property renders cGAS activity highly dependent on both the concentration of cytoplasmic DNA and the concentration of cGAS. So when the cytoplasmic DNA concentration is elevated but still not very high, such as found in TREX1 deficient cells, then the cGAS concentration becomes very critical. If you reduce the cGAS concentration by 50%, it brings it below this phase separation boundary, such that cGAS can no longer be activated to cause disease. So we think that may explain this haploinsufficiency of cGAS in causing disease. And we think this is a very exciting result because it not only provides the genetic proof of concept for targeting cGAS, but it also suggests that partial inhibition of cGAS might provide therapeutic benefits to patients with AGS and other autoimmune diseases. And I will come back to therapeutic targeting of cGAS in the second half of my talk. Okay, so work in the past decades from many labs throughout the world has shown that this cGAS STING pathway plays a central role in many physiological and pathological processes. So this pathway is clearly very important for host defense against microbial infection. The aberrant activation of this pathway can lead to a variety of autoimmune and inflammatory diseases, such as AGS, lupus, and severe COVID. Activation of cGAS by mitochondrial DNA can lead to neurodegeneration, such as Parkinson's disease, ALS, and Alzheimer's disease. We recently showed that activation of this pathway can lead to a non-canonical form of autophagy that regulates lysosome homeostasis. We and others have shown that activation of this pathway can lead to cellular senescence, and this pathway is also very important in anti-tumor immunity. So in today's talk, I will only talk about just one aspect of the cGAS function, and that's its role in autoimmune and inflammatory disease. But before I talk about this, I want to briefly mention the evolution of cGAS. And it turns out that cGAS originates in bacteria. It now joins restriction enzymes in CRISPR as a very important immune defense system against bacteriophage infection. And so work from Philip Kranzul's lab at Harvard and Rotem Sorek's lab at Israel has identified cGAS-like proteins in more than 6,000 bacterial species. And so these bacterial cGAS-like enzymes can make all sorts of cyclic dinucleotides and cyclic oligonucleotides. And the major function of bacterial cGAS-like enzymes is for immune defense against phage infection. And cGAS-like proteins have also been found in archaea, in invertebrate animals, including Drosophila, and has been found in all vertebrate animals, including humans. So this pathway, as well as antiviral function, has been conserved for billions of years from bacterial to humans. Okay, now I want to talk about our efforts in targeting cGAS to treat autoimmune and inflammatory disease, and I'll try to keep this simple. Okay, so in the past few years, there has been a lot of studies that linked the activation of this cGAS STING pathway to inflammatory diseases. This is a table from a review article from Andrea Abrasa's lab. So cGAS activation has been linked to monogenic diseases such as AGS that I just mentioned, Sjögren's lupus, COPA syndrome, SAVI, etc. It has also been linked to more common autoimmune diseases like lupus and arthritis, neurological diseases like Huntington's disease, Parkinson's disease, and Alzheimer's disease, metabolic diseases, fibrosis, and myocardial infections, cancer, and now recently we and others have been shown that this activation of this pathway has been linked to aging as well as aging-related diseases. So it would be nice to have a cGAS inhibitor that has the potential to treat all or some of these diseases. And in fact, we have been trying to develop cGAS inhibitor for many years. After we discovered cGAS, we were so convinced that this would be a good drug target that we started to screen for cGAS inhibitors. And Josh, who discovered cGAS in my lab, joined a startup company called Immune Sensor that I helped started. And after many years of work, we think we have a cGAS inhibitor that's both potent and specific, and it has a good pharmacokinetic properties. It's orally available, meaning that it can be taken as a pill. It has a very good safety profile in preclinical animal testing, the GLP tox study, and has very good efficacy in mouse model of diseases. And so here I will just show you two mouse model of autoimmune disease. And the first one is the TREX1 deficient mice that I showed you earlier. And recall that even if we just remove one allele of cGAS, it can largely rescue the lethal phenotypes of these TREX1 deficient mice. So what happens if we treat these mice with a cGAS inhibitor? So we took TREX1 deficient mice and treat them with a cGAS inhibitor shown in red here, or vehicle control. And the mice treated with a cGAS inhibitor, they can live for a long time, basically phenocopy the cGAS knockout. Measurement of inflammatory cytokines such as CXCL10, interferons in the blood and in the tissues show that mice treated with a cGAS inhibitor shown in red here have greatly reduced production of these inflammatory cytokines as compared to vehicle controls. So that's one model, that's TREX1 deficient mice. And this is another model, it's called the DNS2 knockout mice. And so DNS2 is the major enzyme that digests DNA in the lysosome. So every day in our body, billions of cells die, and this is an enzyme that cleans up DNA from the dead cells in our body to prevent disease. And many years ago, Professor Shige Nagata in Osaka generated DNS2 knockout mice. And these mice are embryonic lethal. The embryos produce so much type of native interferons that kill all the red blood cells. So the embryos die from anemia. And the Professor Nagata tried to rescue these DNS2 knockout mice by crossing them to toll-like receptor knockout mice or MyADA mice, but they couldn't rescue. But they could partially rescue by crossing DNS2 knockout mice with interferon receptor knockout mice. Now these mice are born, they grew up, but they develop very severe arthritis, as shown here. And these phenotypes may be explained this way. So in the absence of DNS2, you have elevated level of DNA that then lead to production of a lot of interferons that cause embryonic lethality. And this can also lead to production of TNF and IL-6. These are inflammatory cytokines that cause arthritis and other disease. So if you take away interferon receptor to block interferon signaling, you can rescue embryonic lethality, but these mice continue to produce a lot of these inflammatory cytokines, they cause arthritis. So that explains why these mice will have severe arthritis. Now, the prediction is that if we take away cGAS, and if cGAS is the major sensor, then you will be able to rescue all the disease. And this is indeed what we found. So here, if we remove cGAS from DNS2 knockout mice, now there's no arthritis. Whereas if you remove interferon receptor from DNS2 knockout mice, they develop severe arthritis. And similarly, if you remove interferon receptor from DNS2 knockout mice, they develop autoantibodies against DNA. And if we remove cGAS from DNS2 deficient mice, then there's greatly reduced autoantibodies. So these results suggest that an inhibitor of cGAS would be potentially superior to interferon receptor blockade. And these results are striking, but also surprising. Because if you think about it, so if you remove DNS2 or TREX1, then this cytoplasmic DNA accumulates. In principle, they will not only activate the cGAS STING pathway, they should also activate toll-like receptor and inflammasome pathways, such as the M2 inflammasome. So one could have argued that if you remove cGAS, you will not be able to rescue the phenotype. But the fact that you can just remove cGAS and completely rescue these phenotypes suggests that in this case, it is the major pathway. And this further validates cGAS as an important drug target for diseases caused by our own DNA. And so now what happens if we treat these DNS2 interferon receptor double knockout mice? They have already developed severe arthritis. So here we took these mice, they have very severe arthritis, and then give them the cGAS inhibitor, shown in red here. Then within 2 weeks, the arthritis score decreased very significantly as compared to the vehicle control. So this is a therapy of this arthritis disease. And the prolonged treatment of cGAS inhibitor did not have any overt adverse events. For example, treatment with the cGAS inhibitor does not affect the body weight in a significant manner. And if we measure inflammatory cytokines such as TNF and IL-6, mice treated with the cGAS inhibitor, shown in red here, have greatly reduced production of these inflammatory cytokines. So these studies suggest that the cGAS inhibitor may be used for treating this inflammatory disease. But what could be the risk? One obvious possibility is a treatment with this cGAS inhibitor might increase sensitivity to infection, such as viral infection. But I think it might be okay. I think a cGAS inhibitor would be similar or even safer than JAK inhibitors, which have already been approved for several diseases such as rheumatoid arthritis. And JAK inhibitors not only inhibit innate immunity, it also inhibit adaptive immunity. For example, it inhibits signaling by several interleukins. Our immune system has a lot of redundancy. So in addition to the cGAS STING pathway, we have RIG-I MAPS pathway that protect us from RNA virus infection. You have toll-like receptors pathways, as you just heard from Professor Okura, the C-type lectin receptor pathway. In addition, we have T cells, B cells, and NK cells. They can protect us from infections. Most humans, I believe everybody in this room has been vaccinated against various diseases, and we all have been exposed to a lot of pathogens. In fact, many human viruses, they have already evolved to evade the cGAS STING pathway. And then as I showed you earlier, we don't have to completely inhibit the cGAS enzyme. Partial inhibition of cGAS may be able to provide therapeutic benefits, and without compromising our immune defense against viral infection. And so if this is still not convincing enough, this is the real data. So here we treat mice with our cGAS inhibitor at a dose that is sufficient to provide therapeutic benefits in TREX1 deficient mice. And then we infect them with herpes simplex virus 1 at a dose that is sufficient to kill cGAS knockout mice. And in the mice treated with a cGAS inhibitor, they are actually completely resistant to HSV infection, similar to the vehicle-treated mice. So that gives us confidence that the cGAS inhibitor will be safe. Now, as I just mentioned, this cGAS STING pathway has been linked to many autoimmune and inflammatory diseases. So if you want to develop a cGAS inhibitor, which disease would you want to try first? And the first clinical trial is very critical. It can only succeed. And so I asked ChatGPT, what would be the good disease indication for a cGAS inhibitor? And it gave me a rank order of these 10 diseases as shown here. These include like lupus, SLE, AGS, and a number of other diseases like IBD, etc. And we have decided to go after the AGS first. This is the disease that I showed you earlier. Although this is a rare disease, but the mechanism of disease is very clear. It's through overactivation of the cGAS pathway. And so the clinical trials of our cGAS inhibitor in patients with AGS is ongoing. The preliminary results are quite very encouraging, and hopefully we can report the results of the study very soon. So in closing, this is the story. It's a very simple one. So we have identified the DNA sensor that turns out to be an enzyme that we call cGAS. And cGAS is activated to a very simple mechanism. It's activated by binding to double-stranded DNA, and the activated enzyme then produces this small but powerful molecule that strongly stimulates immune and inflammatory responses. We believe cGAS is a good drug target, and cGAS inhibitor may be used for the treatment of a variety of autoimmune and inflammatory diseases, as well as age-related diseases and neurodegeneration, such as Alzheimer's disease. In data that I don't have time to show here, we and others have shown that cGAM, this small molecule, and its derivatives have very potent anti-tumor effect in mouse model of disease, but it takes a lot more efforts to translate this into cancer immunotherapy in patients, in human patients. We have also shown that cGAM is a very good adjuvant for vaccine development, at least in mice, and hopefully one day cGAM can be used as an adjuvant in human vaccines for cancer in infectious diseases. So finally, I want to thank the people in my lab who did the work. I have highlighted the work of my postdoc, Josh Chen, and my student, Jiaxi Wu, for the very important contributions in the discovery of cGAS and cGAM. And I have been very fortunate to work with many talented students and postdocs. This is a recent virtual reunion of the lab, and it happened just a few days after the JapanPrize was announced. So we took this screenshot. And finally, I want to thank the teachers and my mentors who have really helped my career, including my PhD advisor, the late Cecile Picker, Dr. Tom and the others, Dr. Michael Brown and Joe Goldstein, my department chair, Eric Olsen, who recruited me to UT Southwestern, and Dr. Dan Podolsky, who is the president of UT Southwestern, who has graciously come to Tokyo for this celebration. And thank you very much for your attention.